Friends, and welcome to this series of lectures in botany again. And today we have an interesting topic to talk about: satellite and repetitive DNA. Well, satellite or repetitive DNA generally refers to the DNA whose base sequence is repeated many times throughout the genome of an organism. It's very common in eukaryotes, accounting for about half of the total DNA, for example, in case of mammals. Its proportion also varies in different organisms and can be, of course, divided on the basis of certain characteristics into various types. While some of this DNA serves as a useful purpose, but a significant proportion of it is of uncertain function and may be treated as junk, or sometimes also referred as selfish DNA, because without having any function it somehow manages its perpetuation. Though used quite often synonymously or interchangeably, satellite or repetitive DNA, they are two actually different things. Let's now talk in some detail about them. What is the satellite and repetitive DNA? Let's first understand satellite DNA. Satellite DNA, in fact, is serially repeated DNA sequences of one or a few nucleotides with a repeat length of up to 250 nucleotides that are not transcribed and are commonly located in the heterochromatin part of the chromosomes. The term satellite DNA was derived from the way in which repetitive DNA is repeated, or rather, when DNA is prepared as a pure fraction, it forms a separate band in the rest of the cellular DNA. Repetitive DNA, so far as concerned, it refers to the substrings of the genome that repeat multiple times and includes, of course, satellite DNA and so-called interspersed repeated DNA sequences as well. The later, interspersed repeated DNA sequences are interspersed throughout the chromosomes in hundreds or thousands of individual copies, each about 300 nucleotides long. Unlike satellite DNA, these repetitive DNAs are generally transcribed. Moreover, different instances of the repeat elements can have slightly different patterns also. It's also highly prevalent in eukaryotes. Those organisms with visible nucleus and well-defined nuclear parts, cell structure, as opposed to the bacteria or prokaryotes. However, the quantum of repetitive DNA varies in different organisms. For example, in case of Homo sapiens, the percentage of repetitive DNA is about 21%. While as in mouse, it's around 35%. In calf, it is 42%. In Drosophila, let me tell you, it is the highest, is 70%. And if we talk about plants, in case of wheat, it is 42%. In case of pea, 52%. In case of maize, 60%. Fungi, such as Saccharomyces cerevisiae, which is in fact a yeast, 5% of its genome is comprised of the repetitive DNA. And as I told you, this is more prevalent in eukaryotes, and as such, prokaryotes have very less quantum of this repetitive DNA. For example, in case of E. coli, it just comprises 0.3% of the total genome. And let me tell you here that repetitive DNA elements comprise a major component of the plant genome. And studying these sequences or these elements is very essential for our understanding of the nature and consequences of the plant genome, size variations between the different species, and for also studying the large-scale organization and evolution of plant genomes. Now, a very interesting question that I will come to is, why to study repetitive DNA at all? As I told you, in case of plants, it's very important for different reasons. Generally speaking, these repeats, they are said to drive evolution in diverse ways through different means. And apart from this, repetitive DNAs are generally not found to have any function apparently, yet they are carried along generations across so many years of evolution. And then third important reason is homology searches need repeat masking. I mean, especially for avoiding explosion of unnecessary results. And also, let me tell you, repeats or repeat sequences, they also contain information about parentage. So there are many reasons for studying this repetitive or satellite DNA. Now the question is, what is the evidence for the existence of repetitive DNA and its exact location in the chromosome? The first evidence for repetitive DNA came from density gradient analysis of eukaryotic DNA. Density gradient analysis or procedure, let me tell you, was first used by Messelson and Stay to demonstrate semi-conservative replication of E. coli chromosomes. The density of 6 molar cesium chloride is about 1.7 grams per cubic centimeter. And if such a solution is centrifuged at a very high speed for long enough period of time, density gradient will be established because of the equilibrium between two things. First, the sedimentation of the cesium chloride to the bottom of the centrifuge tube as a result of the centrifugal force, and second, diffusion of the cesium chloride towards top of the centrifuge tube. Well, when the DNA of a prokaryote such as E. coli is isolated, fragmented and centrifuged to equilibrium in a cesium chloride density gradient, the DNA usually forms a single band in the gradient at a position where the cesium chloride density is equal to the density of the DNA containing about 50% of GC base pairs. And it is important to note that DNA density increases with the increasing GC content, because as you know, GC bonds are much stronger than the AT bonds in the DNA and make it much more tighter and more dense. On the other hand, cesium chloride density gradient analysis of eukaryotic DNA usually reveals the presence of one large band of DNA, usually called as the man band DNA, and one to several small bands. These small bands of DNA are called satellite bands, and the DNA embodied in them is referred to as satellite DNA. The satellite DNA upon isolation and analysis reveals repeating sequences of variable lengths in different organisms. And so far the chromosomal location of satellite DNA is concerned, chromosomal location of satellite DNA have been determined by a technique called in situ hybridization, which usually involves annealing single-stranded isolated radioactive satellite DNAs or complementary RNA sequences synthesized using satellite DNA as a template directly to denatured DNA in chromosome quash preparations and so on. Now, after washing out the non-hybridized radioactive materials, the location of the satellite DNA sequences in the chromosomes are determined by autoradiography. They are usually found in the heterochromatic region of the chromosomes. I mean, non-transcribing region of the chromosomes or inactive region of the chromosomes. But please note that a repetitive DNA sequence will be identified as satellite DNA only if the sequence has a base composition sufficiently different from that of the main band DNA. Only then it is recognized as a distinct band in the density gradient, otherwise it won't be. Apart from these techniques, DNA renaturation kinetics gives a more complete picture of the repetitive DNA complexity and frequency. In fact, the time required for reassociation of particular DNA sequences is inversely proportional to the number of times that sequence is present in the genome. Obviously, highly repetitive DNA sequences will renature very rapidly. Why? Because due to more possibility of collisions between the coherent strands. Well, this repetitive DNA can be classified into various types, starting from, say, the satellite DNA. Mini satellite DNA is another type. Then the other is microsatellite DNA, transposable elements. Even the lines, signs, and other retro sequences are considered as part of the or the type of the repetitive DNA. Apart from these, the high copy number genes, for example, ribosomal genes or histone genes, or even the multifamily member genes, for example, hemoglobin or immunoglobulins, they are the types of repetitive DNA. Let's talk now in some detail about these types. So for the satellite DNA is concerned, as I told you earlier also, it forms a distinct band and its units comprise of 5 to 300 base pairs depending upon the species, and they are repeated usually 105 to 106 times in the genome. And as told earlier also, they are located especially in the heterochromatic region, non-transcribing region of the chromosomes. The best examples of satellite DNA are centromeric DNA and the telomeric DNA. Let me tell you here that there are at least 10 distinct human types of satellite DNA. A single type may be more than 1% of the genome, equivalent to about 3 entire E. coli genomes, you can imagine. And second type is the mini satellite DNA, which is units are 15 to 400 base pairs, averaging about 20, and they are repeated 20 to 50 times. And so for their base pair length is concerned, it is 1000 to 5000, and they are located generally in the euchromatic region of the chromosome, as opposed to the satellite DNA, which I told you is located in the heterochromatic region of the chromosome. The best examples of the mini satellite DNA are DNA fingerprints, tandemly repeated but often interspersed in clusters, also called as VNTRs, variable number tandem repeats. The other type is the microsatellite DNA, which the units of it are 2 to 4 base pairs, mostly of 2 averaging, repeated usually 10 to 100 times in the genome, and they are located also in the euchromatin part of the chromosome. And they are the most useful markers of the population level studies, quite often used there. Apart from these basic types, transposons or transposable elements in eukaryotes are also a type of repetitive DNA. Very famous, as you must be knowing, mase ACDS elements, activator dissociation elements, for example. Likewise, in Drosophila melanogaster, P elements are also the transposable elements. Transposons, as you know, they are also called as jumping genes because they change their position in the genome by way of jumping from one position to the other. And in case of many other organisms, say TC1 mariner elements, it's an exceptionally widespread gene family in case of the animals, ranging from nematodes to mice. Apart from these, as I told you, signs, lines, and the retrosequences. Lines and signs are both the examples of dispersed repetitive DNA. Signs stand for, it is basically SINES. Signs stand for short interspersed nuclear elements, and lines for long interspersed nuclear elements. Signs, for example, ALU family in humans, its average length is about 280 base pairs, and they are repeated about 700 into 10 to the power 3 to 1000 into 10 to the power 3 times in the genome, occurring all over the place, even in the introns. You must be knowing the difference between the introns and the exons. Introns are the transcribing regions, and exons are the non-transcribing regions of the genome. And these signs and lines, they also propagate by transposition. So for the lines are concerned, line 1 is the best example, the copy number of which is 60 into 10 to the power 3 to 100 into 10 to the power 3, usually in the genome, and is a type of non-viral retroelement, a transposon that can replicate and move around the genome by a process involving reverse transcription. The average length of line 1 element is 1.4 kbs or kilobase pairs. Apart from these, as I told you, the other types of repetitive DNA include high copy number genes, for example, very famous and commonly known ribosomal genes, histone genes, then the multifamily member genes, for example, hemoglobulins or immunoglobulins. Well, apart from the different types of repetitive DNA that we have discussed so far, let me tell you that there is one more type of repetitive DNA that makes use of an RNA intermediate. And the best examples that fall under this group include retrons, retroposons, retrotransposons, retroviruses, very famous, for example, AIDS virus, pararetroviruses, and also many other retro sequences. And the fundamental mechanism behind all these types of reverse transcriptase based repetitive DNA intermediates is, the central dogma of molecular biology has been that from DNA is synthesized the messenger RNA through the process of transcription, and from messenger RNA are synthesized the proteins as a result of the translation. But the central dogma, this was changed after the discovery of the reverse transcriptase, because it was found possible that using RNA as an intermediate, DNA can be synthesized in a reverse way. And all these examples are based on that very mechanism, which is very much well known in case of the HIV or AIDS causing virus. Well, let's now come to the functions of the highly repetitive DNA. As I told you earlier also that this repetitive DNA is genetically inactive. It is basically located in a genetically inactive heterochromatin part of the chromosome, the non-functional, or you can say non-coding part of the chromosome. The functions of this type of DNA therefore are almost unknown. And as I told you earlier also that for a long period of time it was considered as junk DNA. However, some functions have been still attributed to this DNA. And these functions include, number one, they are found to, or they have been attributed to have a role in the structural organization of the chromosomes. I mean, they have a structural or an organizational role in the chromosomes. As you know, chromosomes are the carriers of DNA, the basic genetic material. And the way this huge amount of DNA is packaged into the chromosome, and very well packaged, highly efficient mechanism of packaging this is, it is basically made possible through the agency of these repetitive sequences also, apart from the other factors which are there very well known. Then they are also said to have their involvement in the chromosome pairing during meiosis. Meiosis is the process of reductive cell division, wherein chromosome number is reduced to half so as to maintain its number constant generation after generation. And this repetitive DNA, or this non-functional part of the chromosome or heterochromatin, is found to have a role in the pairing of chromosomes during this meiosis, which is an important phenomenon or process. And apart from this, in meiosis, the third important thing, as you know, is the process of crossing over and recombination, which is a fundamental process behind evolution. Repetitive sequences are known to, or they have been attributed to be involved in this process of crossing over or recombination. And through recombination, in fact, the characters of different parents are basically recombined so that the progeny has a different set of characters, and sometimes novel characters which basically help in the evolution of novel genotypes and to withstand other selection pressures. Then the fourth attributed function of repetitive DNA as a whole is the production of important structural genes such as histones, ribosomal RNA genes, or ribosomal protein genes. Well, the validity of all these postulated roles is by and large questionable, and that's why it needs to be ascertained by robust and very well focused experiments. Some people even think that the functions of the repetitive DNA are more than meets the eye and are therefore likely to be unraveled in future course of time. And from that perspective, repetitive DNA with different selective pressures from those acting on genes and evolutionary successful multigene modules or families may show extensive differences in sequence motifs and abundance even between closely related species. Because the coding genes are of course under high selection pressure because they have to produce functional proteins, and this repetitive DNA, the junk DNA, so-called junk DNA, I mean to say, since it is not directly involved in the synthesis of functional proteins, so how it basically withstands selection pressures, or how it basically responds to different selection pressures, is an interesting thing to note in this process. The repetitive DNA in genome is also important for evolutionary, genetic, taxonomic, and many kinds of other applied studies. Let me tell you here, apart from this, or notwithstanding these attributed roles, a few repetitive DNA sequences are known to have very well-defined functions. For example, the telomeric sequences, which are added at the ends of the most plant and animal chromosomes, the telomers, they allow a linear replication unit to be maintained. That's one of their functions. Then the second function is that they protect chromosome ends and overcome also the end replication problems. So these are some of the fundamental roles and very well understood roles that have been found to be played by this repetitive or satellite DNA. The 18S or 5.8S and 5S ribosomal RNA genes or gene loci, clustered at a small number of sites, they encode the structural RNA components of ribosomes. And you can understand how important they are. Mobile DNA sequences such as transposons and retrotransposons, they make up a high proportion of most plant and animal genomes. So these are not only the attributed roles, these are almost very well verified roles that repetitive DNA is known to play. Well, a major class of the retroelements, or these repeated DNA, as I told you, retroelements. These retroelements encode the proteins necessary for their own reverse transcription and integration, and sometimes it represents about 50% of the genome. As a result of their transcription into RNA, reverse transcription into DNA, and integration into the genome, they have a dispersed distribution along the chromosomes. Noteworthy point here is that telomers, ribosomal DNA, and retroelement sequences are all very ancient. In evolutionary terms, I am saying here, and they are found in all animals and plants. And because of this, they are considered to be as early derivatives of the RNA world from the DNA-based organism world. As you know, being the students of evolutionary biology, that earlier world was thought to be RNA world that has given rise to the present DNA world, what we call as. And the role of these repeated sequences, as I mentioned here, is a strong evidence for the existence of that ancient RNA world that ultimately paved way for the present DNA-based world. And in case of plants, so for the survey of major DNA sequences in plant nuclear genome is concerned, satellite DNAs have been found to have varying monomer lengths, but usually 140 to 180 or 300 to 360 base pairs are the frequent, more frequent ones, corresponding to mono or dinucleosomes. Microsatellites are runs of the simple sequence repeats with motifs of 1 to 5 base pairs long, while mini satellites have longer and more complex repeating units up to 40 base pairs long. Telomeric DNA consisting of conserved 7 base pair repeats, and if I exactly tell you, the 7 base pairs are C, 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 T, A, 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 A. I mean 3 times C followed by T and 3 times A. It is added to the chromosome termini by the telomerase, and retroelements amplifying and transposing via RNA intermediates are divided into mobile sequences with long terminal repeats, LTRs, what we very commonly call as, and non-LTRs, lines including then the long interspersed nuclear elements. As I was telling you, lines basically refer to that, and the related signs, short interspersed nuclear genomes. Before we conclude, you must therefore know that repetitive DNA and the genome size. Variation in the gene numbers cannot explain variation in the genome size among the eukaryotes, because the number of functional genes is almost say comparable or not so much variable, but what creates most of the variation in genome size is the amount of non-coding repetitive DNA, mostly transposable elements or other kinds of these repetitive DNA. As I said before also, that the quantum of this repetitive DNA varies across organisms. Transposable elements, they accumulate in intergenic as well as intronic regions. Intronic regions are the coding regions. So in conclusion, we can say that satellite or repetitive DNA is the DNA whose base sequence is repeated throughout the genome many times of any organism. Though it's very common in eukaryotes, but its proportion varies in different kinds of organisms, being more than 50% in some cases, as I told you in case of the Drosophila, and lesser in others. And while some of this DNA serves as very, very useful purpose, it is known to have certain important functions, but much of it or significant proportion of it is said to have no apparent function at all, and that's what's considered as junk. And as I told you, though these sequences or these repetitive or satellite DNAs have been used synonymously or interchangeably earlier, but actually they are different. So satellite or repetitive DNA, which was once considered as a genetic burden, is now perceived by modern molecular biologists as a real gold mine which needs to be very well explored with novel techniques and through new approaches to answer yet many unanswered riddles of the evolutionary biology. So I take leave of you now. Take care and bye.